May God peace, mercy, and blessing upon you, honorable teacher and colleagues, wishing you good health and wellness. Today I will present my seminar which is about halitosis. Halitosis or body breath is a source of embarrassment in social circle. A person having a body breath can be a subject of mockery and may therefore suffer from emotional and psychological distress. So that body breath is considered to be a third most frequent reason for patients to seek the dental care following tooth decay and gum disease. Well, we discussed content which include introduction, classification, epidemiology, etiology, diagnosis, and treatment. At the beginning, let's start with this definition. Breath odor can be defined as the subjective perception after smelling someone's breath. This odor can be pleasant, unpleasant, or even disturbing if not repulsive. If the odor is unpleasant, it can be termed as breath malodor, halitosis, body breath, or Vitrex or. Malodor is the scientific term for body breath. Well, the halitosis is the unpleasant or offensive odor that is emitted from the oral cavity. Classification. Halitosis can be categorized into three main groups: genuine halitosis, pseudohalitosis, and halitophobia. The genuine halitosis can be further subdivided into physiological halitosis and pathological, and the pathological further subdivided into intraoral and extraoral. The first category of halitosis is genuine halitosis. It's obvious malodor with intensity which is beyond socially accepted level. The genuine halitosis is subdivided into first, physiological halitosis. This type of malodor originates in the oral cavity. The main source of origin is likely to be the dorsal surface of the tongue. The physiological halitosis can be caused by food intake like onion, garlic, and some spices. Also by medication like metronidazole and antihistamine. Also can be caused by smoking. This type of halitosis is transient. This means there is no health problem. The same is true for morning body breath, which is caused by decrease in the salivary flow and increase in the bacterial putrefaction during the night. And this is spontaneously disappear after breakfast or after oral hygiene measure. The second type of genuine halitosis is pathological, which is firstly intraoral. That type of halitosis caused by a disease or pathological processes related to the oral cavity, while extraoral pathological halitosis is the malodor caused by disease or pathological processes outside the oral cavity. The second category of halitosis is pseudohalitosis. In this type, there is no obvious malodor perceived by others, but the patient believes that his or he has the oral malodor. The third category is halitophobia. In this type, the patient complains of malodor which is not perceived by others. Halitophobia is characterized by the patient's persistent belief that his or she has the halitosis despite reassurance, treatment, and counseling. Epidemiology. The prevalence of halitosis in the study are controversial. Some of the studies have been reported that the prevalence of halitosis in Polish university students is about 24%, while a study in USA showed that the prevalence of halitosis in patients diagnosed by the dentist is about 40%. Is there a link or a relationship between the halitosis and age or gender? Some of the studies have been reporting that after subdivided the patient into three groups, the first group below the age of 18, the second between 19 and 50, and the fourth above the age of 50. And the study showed that there is an increasing of the oral malodor with increasing the age. Also found the predominance of women more than the men. This may be due to hormonal changes. and these changes will be effect on the salivary flow and protein concentration, thus cause an increase in the volatile sulfur compound concentration and the produce of the such halitosis. Risk factor. The risk factor for halitosis include dry mouth, food intake, alcohol, smoking, and stress. First, in general, all these factors result in increase in the volatile sulfur compound concentration, which is the main contributor of the oral malodor. Firstly, we talk about the dry mouth. The dry mouth have been associated with oral malodor, which can be explained by reduce in the salivary flow, favoring anaerobic bacterial putrefaction of the food debris that remain in the oral cavity after eating. Smoking. A number of studies found that smoking is associated with an increased incidence and severity of halitosis. This is believed is caused either through the cigarette smoke altering the balance of microbial population in the subgingival plaque or through an increase in the absolute number of volatile sulfur compound producing bacteria. While alcohol consumption is another risk factor for bad breath. This is maybe resulting from an oxidation of alcohol in the mouth and liver, producing acetaldehyde and other odorous byproducts. Alcohol may be also cause a hyposalivation and dry mouth. What causes of a bad breath? The etiology of halitosis could be either intraoral, which account 90%, or extraoral, which is at 10%, or a rare halitosis which is caused by dilutional halitosis. Firstly, we're talking about intraoral halitosis. Bacterial role. The bacteria play an important role in the producing of the oral malodor. There is a large microbial diversity found in patients with oral malodor. This has been suggested that there is a complex interaction among the several bacterial species. The most common bacteria involved in the oral malodor include Treponema denticola, Porphyromonas gingivalis, Porphyromonas indentarius, Prevotella intermedia, and Eobacterium. The humidity condition present inside the oral cavity provide an ideal environment for bacterial putrefaction. Peptides present in saliva, gingival tracheal fluid, and blood are hydrolyzed into sulfur-containing and non-sulfide-containing amino acids, which can be further metabolized. The proteolytic degradation of sulfur-containing amino acids by gram-negative bacteria produce a sulfur-containing gases such as hydrogen sulfide and methyl mercaptan, which is the main contributor of the oral malodor. Oral malodor, the unpleasant smell of the breath, mainly originating from volatile sulfur compound that can be either non-sulfur-containing amino acids, which include diamines indole, skatol, polyamide putrescine, propionic acid, acetic, carboxylic acid, and butyric acid, or cadaverine, or either by volatile sulfur-containing compound, which include hydrogen sulfide, methyl mercaptan, dimethyl sulfide. Tongue and tongue coating. Tongue coating is considered the most important contributor to halitosis level. This is properly because of the dorsum surface of the tongue. This surface is irregular, crater-like surface which consists of a complex papillary structure that support the retention of considerable quantities of bacteria. This region also less accessible for routine oral hygiene measures, allowing a greater buildup of bacteria, so that this factor is essential for putrefaction of bacteria and their nutrient and the production of volatile sulfur compounds and the body breath. Periodontal infection. A relationship between periodontitis and oral malodor has been shown. However, periodontally healthy patients can suffer from halitosis, but not all patients with gingivitis and or periodontitis complain from such a bad breath. Several studies have been shown that volatile sulfur compound level in the mouth correlate positively with the depth of the periodontal pocket. The deeper the pocket, the more the anaerobic bacteria and the more number and bleeding tendency of the periodontal pocket. Does periodontitis aggravated by the bad breath? The answer is yes. This is because, firstly, hydrogen sulfide and methyl mercaptan facilitate the penetration of lipopolysaccharide into gingival epithelium, inducing inflammation. Also, volatile sulfur compounds aid bacterial invasion of the connective tissue by their toxic effect on the epithelial cells. Second, methyl mercaptan hinders epithelial cell growth and proliferation. And also, third, results in embedding of the wound healing. Some studies, however, have been shown that when the presence of tongue coating, the correlation between periodontitis and the oral malodor is much lower, so that the tongue coating remains a key factor for halitosis. The prevalence of tongue coating is 6 times higher in patients with periodontitis, and the same bacterial species associated with periodontal disease can also be found in a large number on the dorsal surface of the tongue. Other intraoral causes include caries, orthodontics, dentures, oral ulcers, tooth extracted, dry socket, pericoronitis, and acute necrotizing ulcerative gingivitis. Extraoral halitosis. Extraoral halitosis. Extraoral halitosis are responsible for 10% of the overall body breath. This halitosis can be originated either from respiratory system or from gastrointestinal system or from metabolic disease or can be originated from the drugs. Firstly, halitosis originated from respiratory system. Respiratory tract infections such as pharyngitis, sinusitis, the presence of foreign body in the nose, and tonsil stone. All these infections result in accumulation of the debris and bacterial putrefaction resulting in the bad breath. Ozena, which is caused by Klebsiella ozeni, is a rare atrophic condition of the nasal mucosa with the appearance of the crust that causes a very strong breath malodor. Bronchi and lungs. Pulmonary causes include chronic bronchitis, pneumonia, pulmonary abscess, bronchial carcinoma, and carcinoma of the lung. Also result to the bad breath, and the prevalence of such condition require an early diagnosis. Secondly, halitosis originated from the gastrointestinal system. Gastrointestinal system are rarely responsible for the body breath. They are responsible for only less than 1% of the breath malodor. For example, Zinker diverticulum, which is a hernia in the esophageal wall, allowing accumulation of the food and debris and thus putrefaction of the bacteria. Secondly, regurgitation esophagitis, which is ulceration of the mucosal lining of the esophagus by acidic stomach content flowing back because of the improper functioning of the sphincter. Also, H. pylori infection have been shown to produce hydrogen sulfide and methyl mercaptan, a finding suggesting that this microorganism can contribute to the development of halitosis. Third, halitosis originated from metabolic disease. Patients with liver disease may be acquire a breath which is a sweet, musty, or the breath termed as bitter hepaticus, while patients with a kidney disease may have a fishy smell. This may be occur as a result of accumulation of dimethylamine and trimethylamine, which result to the such body breath. Patients with uncontrolled diabetes result to a rotten apple smell. This is because of accumulation of the ketone body. While trimethylamininuria, which is enzymatic defect caused as a result of failure in the transformation of the trimethylamine to trimethylamine oxide, which results to the fishy smell. Fourth, halitosis originated from the drugs. These drugs include chemotherapy drug, metronidazole, biphosphonate, antihistamine, acetylmethylphenidate, dimethyl sulfoxide, disulfiram, and nitrate and nitrate. Delusional halitosis. In addition to the conventional halitosis, about 5% of the cases are termed delusional halitosis. These situations where the patient complains of persistent malodor, but there are no subjective or objective signs from examination and further investigation. These can be either the patient feeling that he has a such problem, which is halitosis, or either the patient afraid from develop such a condition, which is called halitophobia. These cases are very rare and should be discarded when examination of the patient in the dental chair. Physiology of the malodor detection. The breath of person contains about 100 different molecules. The perception of molecules depends on the following factor. Olfactory response. The odor itself can be pleasant, unpleasant, or even repulsive. Threshold concentration. Each molecule has a specific concentration before it can be detected. Odor power, which is the concentration that is necessary to increase the odor source. The volatility of the compound. The malodor molecule only expressed when they became volatile. The substantivity, which is the capacity of the molecule to stay present and thus remain the cause of the smell.